All right. Welcome everyone for joining us on this last day of Genesis Week. And not the least, because we have a really exciting program today, starting with a very exciting workshop focusing on the Japanese startup ecosystem in life sciences. So in this hour, you will have a really good overview of opportunities of collaboration with Japanese life science ecosystem, and then a pitch of companies that have also very exciting science to show you and showcase to you. Obviously, there will be time for questions, either to our host about the life science scene in Japan, but also to the companies. And obviously, companies are also registered on the Genesis app. So if you want to reach out to them directly, feel free to and ask them question or organize meeting. So as you heard from the beginning, this meeting is recorded, which is also a good opportunity to get back to it or to share with colleagues who would be interested in the Japanese life science scene. And that's it for me. So I'm going to leave the floor to Masahi. It's going to be your chair today and introducing everyone and obviously taking questions. Sorry, one tiny point. I said I was done. If you have questions, if you can drop them in the chat box, so then we pick up on them, that will make it easier for conversation rather than everyone talking at the same time. Thanks very much. And Masahi, over to you.
Cool. Thank you very much, Aline. Yes, how are you all? Good morning, UK, and good evening, actually, for those joining from Japan. And thanks for joining the innovation workshop. This is the last day on Friday, and co-organized and sponsored by LinkJ, Life Science Innovation Network in Japan. And LinkJ and One Nucleus engaged MOU to exchange information and network between mainly in the UK and Japan as leading ecosystem organizations. My name is Masashi, supporter of LinkJ, and I am acting as like ambassador because I'm based in the UK. I was working for Oxentia, Oxford University Global Innovation Consultant firm. So we nominated 5 leading startup companies this year. And the topics are very diverse, with having a cell biology, cell therapy, vaccine platform, and new drug development platform using a space environment. So hope you enjoy the session. So before we crack on, I will invite the CEO of LinkJ, Mr. Aki Soyama, for the opening remarks. Soyama-san.
Okay, Masashi-san, thank you very much. Hello, everybody. I'm Aki Soyama, President and CEO of LinkJ. So LinkJ is a non-profit organization. Its official name is Life Science Innovation Network Japan. L means a wide range of life science. I means to support and promote innovation by creating N&K network or ecosystem, both in Japan and abroad. That means J. So we have more than 500 memberships, and our history is 5.5 years, and we have hosted a lot of events and programs to support this innovation. And today, 5 promising Japanese startups will make you pitch for everybody. So I hope everybody will enjoy this session and to connect to those promising 5 startups later. Thank you very much. So Masashi-san, please.
Great, thank you, Soyama-san, for the introduction. So let's crack on. So first speaker is Yosuke Matsumiya from Luca Science. So Luca Science is a pioneering company for mitochondrial therapy. The current focus on them is on the pregnancy-related disease, but having a very cool platform technologies can be used for many unmet medical needs. So all for you too, Yos.
Great, thank you, everyone. Can you hear me? Just making sure. Excellent.
Yep, I can hear you.
Really, thank you to Dr. Matsunaga, LinkJ, and One Nucleus for this opportunity to present. So I'm Dr. Yos Matsumiya, and I'm the Managing Director of Luca Science UK. So I thought start off with a journey in pregnancy, as I'm a doctor who used to look after pregnant women. So I just want you to take a moment to imagine. So some of you may have experienced this already. So you, your partner, your relative, or your close friend is pregnant. And actually this was when my son was pregnant, and he's now 6 years old. So this is a very exciting time. This is a typical growth chart that you get where you plot how big your baby is, and you've had your 20-week scan, and that is normal. At 28 weeks, the healthcare professional is a bit worried about the growth, and you have an ultrasound scan on the smaller side. So you have another scan 2 weeks later, and this is a specialized ultrasound scan. And the doctor tells you you have something called severe RUGR. So the question is, what is RUGR? So RUGR stands for intrauterine growth restriction. Some people also call it fetal growth restriction, FGR. It affects 10% of all births worldwide, but is present in 50% of all babies that die during pregnancy. So this equates to 10 million babies per year worldwide, and over 1 million stillbirths per year due to RUGR. So actually, that's 2 babies dying of RUGR per minute. So I've talked for 2 minutes, and that's been 4 babies who've died because of it. Even if they survive, they are born prematurely, affecting morbidity. So the average pregnancy is 40 weeks. RUGR is usually less than 35 weeks. And this has long-term risks of metabolic cardiovascular disease, as well as neurological development, such as IQ. This has a huge economical burden. So just in the UK alone, for better RUGR detection and treatment, it has costed at £56 million per year. And this does not include the cost of the neonatal ITU bed, which will be required if your baby is born prematurely. So what causes RUGR? So this is the lack of energy being provided by the placenta. So this is a placenta connected to the baby. This is incidentally me delivering the baby. The involvement of mitochondria is very well documented. And something that is quite clever in science is that the baby's mitochondria is 100% the mother's mitochondria. And this is one of the main messages I want to get across, is there is no treatment currently for RUGR. All you do is you watch the baby keep on growing, and when the growth stops, you deliver the baby, usually by cesarean section. And the reason there is no treatment is so far we have been unable to isolate mitochondria, we are unable to store them, and we are unable to direct treatment to placenta until we came along. So Leucoscience. So we are a preclinical mitochondrial platform company established in Japan in 2018. We have headquarters in Tokyo, research lab in Sapporo in the north. We raised about $10 million US for Series A last year. And we also got the equivalent of an NIH grant last year for COVID as well. We set up a UK subsidiary, and the main reason is to focus on the RUGR pipeline, working in collaboration with the University of Oxford, which is where I'm based. And we are currently actively looking for local funding and manufacturers. And globally, we are planning to raise further funding by second quarter next year. So we are able to isolate functional mitochondria, deliver them, and we want to use it as a biopharmaceutical platform. And as you can imagine, there are lots of potential pipelines, including space, which I know someone is talking about later. But we've tried to focus on 4 aspects. So MI, lungs, RUGR in the UK, and T-cell exhaustion as well. And we have a lot of global collaborators and advisories. So I'm not going to go through all this, but lots of people ask, how do you know our mitochondria are functional? And these are all the assays we have done to show that we can keep them very functional all the way up to over a month. And just quickly, an in vivo experiment that we've done. So basically we caused a heart attack in a pig, and we injected what we call Q. Q is the functional mitochondria that we use at the time of reperfusion, and this showed significant improvement in heart function, even after 15 minutes, sustained after 1 week. And in mice, we have data showing this lasted for up to 1 month. At Oxford, we have successfully isolated the mitochondria Q from mother's blood. And we have a very clever targeting system. So the far left is the placental tissue, and the red is our target. And you can see in the other cells, it doesn't go in very well without it. So just going back, I'd like you to imagine, very much like some of you, to become part of this life-changing journey with us. So less on the left and more on the right. It's a baby I delivered in Uganda when I used to volunteer there, and discovering a new frontier in organelle therapy. Thank you very much.
Excellent. Thank you very much for the presentation, Jos. So if the audiences have got question, please feel free to put in a chat box so I can just digest. For the time being, I have one or two questions, not technical, but for thinking of the UK-Japan exchanging, and you are quite advanced with having a UK function and a legal entity. What has been changed after establishing a corporate in the UK as a Japanese startup company? Yes.
Yeah, thanks, Dr. Matsunaga. So the question is, what's changed since setting up a UK subsidiary? So the first thing is having a base here. It's a lot easier to collaborate with, say, the University of Oxford, who are our main research collaborators, using their laboratory. We have access to some amazing staff, both at a sort of technical lab level, but also getting to know people from business development level, and actually getting to know people like you, Dr. Matsunaga, and Oxentia has really been, important for us to know how the system works in the UK. We've managed to reach out to investors, and also we can apply for grants within the UK, and you can only do that if you have a UK company. So actually having a subsidiary in an area that you really want to focus on has opened up our opportunities, both at a research level, but also moving on to manufacturing and investors.
Cool. What is your next demand?
So I think demand in something like this comes sort of both ways. One, we want to keep on producing results. And for that, we need not just the knowledge, but we do also need the funding. So we are always looking out for investment, and locally as well, because we have a UK subsidiary. And obviously those who are interested in what's happening at a global level have the opportunity to invest globally. And we're very lucky today, actually, that our CEO of the company, Rick Tsai, has joined us, and he's currently joining us from LA.
Cool. Thank you very much. I've got some questions, but I will have to go to the next one because time is quite limited. So maybe after the session, if we got time, we'll generate all the questions. So thank you very much, Jos. Great presentation. So next one is Aku-san, can you just speak? Sorry, we still cannot get your voice. No, sorry. So maybe we can change the switch. Hello, yes.
Oh, now we can hear you.
Hello.
Yes, good.
Okay,
good.
thanks. So,
Right.
Hold the iPhone.
Good.
I'm
All right.
very
So we keep the order. So second speaker is the Takumitsu Akutsu from Curips, and Curips is an iPS derived cardiomyocyte seed technology plus allogenic cell therapy company. One of their focus is a very well-known scientist in academia in Japan. I know that person very well, and they are challenging to the global markets. So all for you to Akutsu-san.
Okay. So can you see my screen?
Yes, I can see your screen. I can hear
okay.
you very well. Thank you.
Okay, thanks. So I'm really happy to be here. And so I want to introduce our company, Curips Inc. This is Akutsu. And so I'm really excited to introduce our product. This is our first product, iPSC derived cardiomyocyte patch. It's a bleeding. And so Curips, it comes from the Cuore, heart in Italian, and iPS. It's Curips. Our vision is to save heart failure patients with a regenerative medicine and cell therapy technology. So these are facts of the heart failure. as you know, so the heart failure is the most cause of death in the world. 26 million, this is a heart failure patient in the world. The 1.3 million, this is a NYHA class 4. It's estimated 5%. It's a very severe heart failure patient, 1.3 million. And the 5,000, this is the heart transplant conducted in 1 year. It's around 5,000. But on the other hand, 50,000 patients are waiting for the donor for the heart transplant. This is a big challenge. And also the cost side, the cost of the heart transplant in US is around 1.4 million. It's a very, very huge cost. So now, we created some solution to, solve this problem. So our Corvus, we are the company using iPSC-derived cell therapy for heart failure. And so we have a first iPSC-derived CM product from Osaka University. And then, so we already have a stable and robust manufacturing process and facility for the off-the-shelf product with Japan quality. In the end, so we have a lot of partnerships with diverse corporate and the next pipeline for the future growth. So about our company, so we have established March 2017. So our CEO is Takayuki Kusanagi. And we already have 3 facilities, Tokyo and Osaka. And so these are some topics from our side. So we opened our manufacturing facility in Osaka last year. And so we conducted the 3rd and 4th private placement. It's around $33 million. And so we are really, excited. So the Professor Yoshiki Sawa is appointed as our new CDO. So one technology from the best academia, Osaka University. And so we have an exclusive right to use technology and know-how from Professor Yoshiki Sawa's group. Professor Yoshiki Sawa is really great. And so he's a cardiovascular surgeon and scientist, both sides. And so he led as a head of the cardiovascular surgery, Osaka University. And also he's the president of the Japan Society of Regenerative Medicine until this year. He has a lot of experience, achievement, and insight for this field. And so we are really happy. He's a founder and CTO of our company. He really knows the bench to bedside and he loves science. And so this is our product again. So we have an iPS cell from Kyoto University. And so we differentiated iPSC CM. And then also we created this kind of patch with seed technology from the Tokyo Women's Medical University. So this is a great news from the Osaka University. So they conducted the first in-human clinical trial from January 2020. So this is a doctor-initiated clinical trial for the ICM. And so they already conducted 3 patients in phase. So now they evaluate these patients, and so they will soon report the clinical, evidence from, officially. But we heard so all the patients, so NYHA class is improved, and also they discharged from the hospital, and so they return to the workplace. This is great, and so they improve the QOL, and also I believe so we can, contribute to the patients all over the world. And so for the commercialization, so it's a stable and robust protocol for the safety and scale. And so our technology with Osaka University, we can derive cardiomyocytes from iPS cells over 90%. And so large scale cell culture is over 1 billion. It's huge. And so as you know, the iPS cell technology is really key. So purification and removing anaplastic cells, and then tumorologic control technology is really key. And additionally, so there's no arrhythmia case so far. It's a very safe approach for the patient now. And so this is our market assumption for the first product. It's a very easy one. But and as I already mentioned, worldwide population of the heart failure is 26 million. And the NYHA class 4 is around 5%. It means that 1.3 million is our opportunity. And almost last slide, so we have investors. So as you can see, so there's so many venture capitals. But on the other hand, so we have a lot of corporate investors. As Daiichi Sankyo, Mitsubishi Corporation, and Terumo, Fujifilm, Asahi Intech, this is a capital company. And so now, so we leverage this kind of collaboration to create a second and third product as a new pipeline for the future growth. But so our aspiration is go global. So we are really, looking forward to discuss with a global partner, and so the full future growth I'm really looking forward to discuss if you want some interest for us. So our company again, so it's the same slide. So we are the Quoripus. So this is the end of the presentation. Thank you for attention.
Thank you very much, Akusan. It's great presentation. Thank you. So,
Yes, thank you.
Because Professor Sawa is quite well known in Japan for the cardio clinical studies, and your last message is to go to the global market. So, what is the message to the audiences for the? Do you want to have some clinical collaboration or grant or the investment?
Yes. So every opportunity. So mainly, so we need some mega pharma so collaboration with to go to the global side, and so we believe so our technology is already advanced, and also we're number one in this technology for especially iPSC cardiomyocyte area. So, but on the other hand, so we don't have any experience for the global side. So it's already great to connect with a global company, and so we want to understand each market. This is my message.
Sure, great, thank you.
Thank you.
Yeah, I was in the IPS company before in Cambridge. So, do you plan to generate anything like a disease models, or do you consist on the healthy wild type?
Okay, good question. So another our project. So now so we are thinking about some model, and also we're working on that. So it's not open. So, but we're working a lot of projects with Dr. Sawa.
Great, thank you. Because you talked to something like a second, third project. So I just guessed.
Yes. A lot. Projects. After the NDA, so we can talk.
Sure, great. So if the global the alliance, you can target from the audiences, maybe you can have free to have a chat with this company. Thank you very much. Great presentation.
Thank you very much. Thank you.
Right. Go to the next one. So next one is Kazuhiro Matsuda from M Biotechnologies. So they are the mycoplasma specialist company in Japan. And they detected specific antigen and finally succeeded to do chemical synthesis and to be applied for the new vaccine platform development. And maybe he will point something for the new like a concept for the relationship with long COVID, and which will be the hard topic at this moment. So I will hand over to Dr. Matsuda, please.
Thank you very much, Matsunaga-san and Sayama-san, and organizing committee for Genesis 2021. I, Kazuhiro Matsuda, is CEO and founder of M Biotechnology. We are an R&D company based in Japan with having very strong patent portfolio and platform technologies, which may change the concept behind mycoplasma infectious diseases, MID. What is MID? MID has strong correlation with many major diseases and orphan diseases, even with the long COVID syndrome. However, tend to be diagnosed just as cold. MID is a spectrum of infectious diseases with a large unmet medical needs, not only asthma, COPD, such as CNS, vasculitis, cardiovascular, kidney diseases. However, it is overlooked with the current diagnosis, which can only detect in quite limited disease types, such as pneumonia. The market size is 1 billion worldwide already. However, using our innovative diagnostics, we could reveal clear landscaping of MID. Then the market size is estimated $10 to $20 billion. the market size of vaccine is expected 10 to 20 billion only for mycobacterial pneumonia, which is same as pneumococcus vaccine market size. Therapeutic antibody market size is also expected same as those. After 30 years of basic research, cutting-edge technology, we have prepared diagnostics and drug candidates. Because Mycoplasma is the smallest bacteria and was difficult to isolate specific antigen, we have detected highly specific glycolipid antigen on Mycoplasma membrane, elucidate their structure and succeeded in chemical synthesis. These patents covering diagnosis, prevention, and therapy have been approved worldwide. The diagnostics is revealing the landscape of MID spectrum. The chemically synthesized antigen is applied to make innovative and very safe vaccine products. Also, we succeed to generate antibodies using the core platform technologies. These products lead us to ideal treatment that identifies the cause of an intractable disease that couldn't be cured before. This slide shows the business model. Develop diagnostics and concept is global life science vendors. Develop a vaccine manufacturing platform is a global pharmaceutical company. Antibody production and portfolio development with selected business partners, and increase market presence and concept through global business development partners. This LEAD Super City project is global consortium with government and growth strategies, leveraging global funding for clinical research. We hope mycoplasma infection, which have already pandemic, global infection then require public health support as well. What is the co-infection rate of mycoplasma in COVID-19 patients? A screening of COVID-19 patients evaluate the status of mycoplasma infection with serum samples. A screening of patients being treated for long COVID in the context of chronic mycoplasma infection treatment or mycoplasma infection improves symptoms for the time course of mycoplasma symptoms progression. MID PRISM are able to solve those problems and to determine the therapeutic effects. MID is a people disease is required support from global stakeholders. The UK is advanced society investigate the long COVID effects. We are willing to collaborate with NIHR long COVID research, UK, EU universities, clinical and central laboratories. Global collaboration, we propose directions for public health, health industries and ecosystems, including to global infrastructure. Milestone discussions undergoing with global pharma companies, ongoing joint research project in National Institute, Japan, US, UK, and Europe, and also mass production of synthetic glycolipid antigen with European partners. From preclinical to early clinical study to see safety and efficacy POC, GLP GMP chemical synthesis is of the antigen, it needs $4 million. Then CMC and preclinical study, $6 million, and phase 1 study, $10 million, totally $20 million for the early POC. Then phase 2-3 study for orphan drug designation, totally $50 to $100 million. We are looking for funds and investment for those. And biotechnology is looking for technical and business collaborations from the global markets. Please call us to open the door for a new era of precision medicine. Thank you very much for your attention.
Great. Thank you very much, Matsuda-san. Yes, that's brilliant technologies, and congratulations on the success of synthesis of a special antigen. So am I correct? Your business order is to start like a special detection diagnosis system, then go to vaccine development, then finally apply to the antibody production. Am I correct?
Yes, yes. We are totally, so we are thinking about the precision medicine for the MID, for the comprehensive maybe medicine we are thinking about. And we have antigen. So this antigen technology is a world first and leading technology. So based on these antigens, we can start. We are already start for the diagnostics to seeing the antibody test, so we can see these patients or epidemiological study and thinking about the precision medicine. And at the same time, we already established the vaccine candidate and therapeutic antibody candidate to develop. So targeting these patients and seeing the pathogenesis of those. And then at the fastest one is a vaccine development. So we already known GLP level of the safety and efficacy. So now we are developing for the GLP-GMP antigen synthesis. Then vaccine CMC, so very simple. And then clinical development for phase 1, preclinical study and phase 1 study for 20 people to the safety. This is the fastest step for us at currently. But at the same time, if we have partners, we can do these comprehensively.
Cool, thank you. When do you expect to launch the vaccine in the UK, for example? Because for the COVID, the vaccines are like always key. And when can we expect to have your vaccine?
Yes, that's very important, interesting, and very attractive. And because to think about the infectious disease and see epidemiology, and so developing the COVID-19 vaccine, so UK is maybe very fast country to develop to do. So also they have courage to seeing these patients COVID-19 for the Oxford University that they are named long COVID. And so using our diagnostics and seeing the patients, and so super infection on my MID, so we can reduce the disease burden of those. Then maybe at the time the importance of the vaccine is understood. Then also UK have NIHR, such kind of UK fund, and together with UK companies, very attractive and interesting to thinking about those collaborations.
Cool, cool. Thank you very much, Matsuda-san. It's beautiful. Yes, we finger crossed for having a
Okay.
Thank you very much.
Thank you very much.
Thank you. So we can go for the next. Fourth speaker is Hide Takahashi from Purec. And Purec is a MSC, a specialist company, and being able to produce 10 to the 12th numbers of pure, pure unstable cells suitable for cell therapeutics. And they're now focusing on the major bone metabolic disorders. Hide will have another idea to develop to the new field. So all over to Hide, please.
Hi, thank you very much for this great opportunity. I am Hide Takahashi, CEO of Purec. Purec is a cell therapy company with a special MSc called REC, especially for bone regeneration. We plan to have very fast in human clinical trials next year. Today, I really look for potential development partners and investors. Our first target is called hypophosphatasia, a rare bone formation disease, like 1 in 100,000 population. There is no cure at this moment. Unfortunately, many baby patients just die. Surviving patients are facing outrageously expensive drug claims, like $2 million per year for lifetime. Can we allow this? That was a question of New York Times 2 years ago. Do we have any other options? That was a question of a doctor in Japan 15 years ago. Dr. Taketani, our friend, made a historical human trials for hypophosphatasia with MSC. Actually, his experimental trial was successful, but the Dr. Taketani called it partial success because the baby bones formation became stopped after 18 months. It was the limit with the normal MSC. Dr. Taketani at that time just wished if he had had any better MSC. Actually, regular MSC do not grow at commercially viable level usually, and their differentiation abilities are very limited to be the source of the other human body organs. To make it worse, regular MSCs are often found clogged in lung areas. On the other hand, our product, REC, conquered all the challenges associated with regular MSC. First of all, REC, as it's named, can rapidly expand beyond the 10 to 12 within 12 weeks from a single cell. REC can show clearer differentiation ability compared with the normal MSC, especially for bone tissues. More importantly, REC is clog-free in its migration in the body. So, in short, in business world, low-cost production, effective therapy, and safe therapy. We are ready to apply these unique features of the cell therapy by REC for many clinical applications. We are already getting results not only in vitro, but rather also in vivo, especially for bone regeneration field. First, in an animal study of hypophosphatasia, we observed clear engraftment of REC even in the mouse bone to provide human osteoblast, unbelievably. And we already obtained the green light by our authority in Japan to start the initial clinical trial next year. Also, in another animal study of spinal canal stenosis or disc herniation, in other words, regenerated disc cells were observed with REC treatment. Then we are going to file IND early next year to start another clinical trial later next year. REC is ready for production. Our company has already established a whole process of production to provide REC to clinical trials. Also, we have a multi-layered IP protection. One process patent has been already issued in Japan, Singapore, and Australia. And also we have another process patent and also 2 use patents filed. Our company has a quite an experienced and balanced management team. 2 are from medical community, including our very founder, Dr. Yumi Matsuzaki. The other 2 are from more business side. Actually, I am from the business community, and I had a chance to serve for 2 different drug development startups, one in US and the other one in Japan. Among the vast and broad market potential for the MSC treatment, we plan to cover smaller indication with REC by ourselves, like hypophosphatasia. While larger market will be covered with the licensed partners, like spinal canal stenosis. At this stage, only these 3 pillars of the indication, market size would be more than $23 billion. And if we can scoop up just 1% or 5%, whatever, that would be gorgeous as a small company like us. We already have a clear picture of how to cultivate the targeted field in Japan, at least. On the other hand, we still have to prepare for overseas reach, especially in 2 areas. First, development partners, and second, investors. I have to add one thing because I saw some CMO company's name in the audience. So we also need to say help for the manufacturing area because we already got the grant for Japanese production, but to make it say GMP equivalent for say European community or US, we have to streamline the process. So we need your help too. Hope I can set a deeper dive with the potential partners and investors after this presentation. Thank you very much.
Oh, thank you very much, Hide. It's a great presentation. I have some questions. And
Okay,
if
shoot.
the yeah, thank you. Yes, so I nearly forgot. So what type of like a partnership do you want? Because I feel like you can have two business models. One is a simple like a distribution models because you can have sell product and you can use a distributor, etc., to reach to the pharmaceutical company. Or do you want to focus more on the collaboration with a big pharma?
Of course, both any kind of collaboration would be possible, but we are looking for partners who can actually develop some clinical trials, I would say cells for some specific indications, which could be the same indications as we are doing in Japan, or other disease targets are also welcome to discuss. Yeah.
Cool, thank you. And also for the hypophosphatasia, so you are starting doctor-led clinical investigation. But my feeling is that if you do everything in Japan, it's a bit slower. And if you shift the clinical study focus to the US or UK or Europe, that can accelerate your clinical study development. What is your opinion?
Yeah, we have, say, two different school of thoughts. One, Japan has a special, say, designation program for the, say, pharmaceutical drugs application, like accelerated program. That is quite, say, advantageous, if I may call it, compared with other countries, especially for the regenerative medicine. So we may utilize that advantage to get accelerated approval in Japan. That's one idea. But the other idea is to seek for the bigger market and to seek for other grants. We may have to go into the, say, other country, like the UK or other countries. But to do so, I need the help for GMP preparation, which is a challenge. So I really would like to seek for other type of partners in manufacturing too. Yeah.
Great, thank you very much. Because I am trying to recall like a soft landing to the other countries, including the UK.
Yeah.
So doing clinical studies or further investment. So I try to help your development.
Great.
Right.
Any help would be welcome. Yeah. So
Thank you very much, Hide. It's a great presentation.
Okay. Thank you very much. Yeah.
Thank you. Good. Thank you for keeping the time, which I like the Japanese speakers keep always on time. And the final speaker will be Shun Yamaguchi from Space BD. So this may be a quite different topic. So innovative drug discovery supporting a company using a microgravity in the space, so-called GFDD or gravity-free drug discovery, which sounds new for me. And then the people can obtain very pure crystal material using that conditions. So I'm quite interested in your presentation. So all over to Shun. Thanks.
Hey, thanks. Good morning to everyone and a good evening to Japanese. I'm Shun Yamaguchi from Space BD. I'm the final presenter from Japan and from a space company. I'm talking about innovative and proven space drug discovery and our innovative gravity-free drug discovery GFDD platform. So I'll get straight to the point. Next slide. So what is our solution to the problem? So our solution is a very generic technology platform to tackle the huge cost in drug discovery. Then we realize faster and more efficient process in drug discovery. This is not difficult to understand, I guess. So the question is how. But before going deeper, what is drug discovery process in general? This is very conceptual, but mostly drug discovery is optimization of proteins and candidate compound interaction using 3D structure data of those molecular and computation. So as you can see on this slide, so it's a combination of target proteins and searching the drug candidate, whether to fit or like how to fit and how to like edit. Then in this context, white space experiment is beneficial. So the answer is here. Through space experiment, we can obtain super high resolution 3D structure data of proteins. For example, like better than 1.5 angstrom. As you can see on the right hand, the right hand is a space experiment. So you can see, I mean, the resolution is 1.3 angstrom. Compared to the ground experiment, you can see it's a little vague. So you cannot determine the structure precisely. But thanks to the space experiment, you can get a more precise 3D structure data for the computation and simulation. So how such super high resolution structure data is obtained? So such data comes from those high quality crystals that can be uniquely obtained in space. So again, you can see the space experiment result on the right hand, and while the ground experiment is left hand, this is the crystal. And as you can see, we can get a higher crystal thanks to the space experiment. Then using this crystal, we do the X-ray crystallography to get the 3D structure data. Then the higher quality crystal in space. This is a key for using space. And in microgravity in space, there's no convection from density differences. So the protein crystalline can grow in a quiet and stable environment. But this is how this space platform works. So as you can see on the left hand ground experiment, there's a convection. So crystals don't grow in a very stable environment. But on the other hand, the space experiment, you can get a very stable crystal. So this is how the platform works. So to sum up, again, the better crystal in space can generate better 3D structure data of molecules, then lead to more efficient drug discovery using the computation and machine learning, the AI, etc. Then this is our track record. So actually, this sounds quite new, but actually we have already 20 years operation and development partner with JAXA. So JAXA has, launched more than 1,000 proteins to the space, and those successful results already obtained. And this is some of the track record in terms of resolution. That's what I showed in the other side. So this is already publicly open. You can easily look into those theses in the website. Yeah. Then I guess you are now interested in this exciting platform. Then the next question would be when the launching opportunities. We actually secure regular access to the space thanks to our partnership with JAXA, Japanese Space Agency. So then our next flight is 2022 summer, coming very soon, and still continue regularly, two or three flights per year, at least till the 2024. So this would be the good opportunity for you. And in then, well, if you are not familiar with, this is all about the protein crystallization actually. But if you are not familiar with those techniques, we can provide you with the end-to-end service, not only the space experiment. So as you can see, we can start from number one on the ground experiment with our crystallization experts working on this 20+ years. Then we can, of course, help you going to the space. And even after return from the space to the ground, we can deliver those X-ray crystal analysis on the ground. So let's explore the space together. Thank you very much.
Thank you, Sean. I enjoyed. It's very nice presentation. Thank you. So, yes, crystal to angstrom to space. This is a very big order of magnitudes. And if I can start comment from someone living in the UK or Europe, the space service is normally people can imagine like a NASA or American based. Is there any similar services, or what is your competitive landscape if you do with JAXA in Japan?
Oh, are you asking me?
Yes. So what is like a unique point if the UK candidate, your client, potential client, want to use your service? What is the difference between the competitor?
Sure. So yeah, there are two major points. One is that we can use the 20+ years technology developed by JAXA and our partner. So compared the uniquenesses of JAXA that they are very focusing on these experiments among other space experiment techniques. So that I believe that would be the strong point of JAXA's technology. And the other one is that, like, as I show here, we have regular access. We have secured already regular access to the space. So it's not like you apply and fail, and you apply and fail. That's not like that. So we can already secure the opportunity in partnership with JAXA. So you can plan very firmly with our experiment.
Great, thank you very much. I like the concept because the rocket, the discovery is something used to be like a more like a war or to show the country dignity, but now can be used for the drug discovery or much safer applications. I really appreciate. And also because AI discovery, drug discoveries are getting popular. So we got some hit candidate on using the PC, for example, but some people go failure to do the actual chemical synthesis.
Sure.
So you are supporting on that kind of like a bridging process. Am I
right? Definitely.
Sure, thank you. Any messages to the UK audiences?
Oh,
what's your demand?
Please join anyway. This journey, exciting journey, that you know what you can get.
Cool. Thank you very much. Thank you, Shun. It's a great presentation. Right. All right. So I enjoyed the 5 presentations. Thanks for all the speakers. And I left a couple of questions, and 2 are for Yos for the Luca Science. If you can show your face. And I got 2 questions from David. First one is this is presumably a very sensitive area for experimental treatment in a clinical trial. And what is your clinical trial plans? Can you comment?
Yeah, thanks. So actually, in the background, I've messaged them both back, but for everyone's sake, I shall say what I've answered. So it's a very good question. So like you said, the question is about clinical trials, isn't it? What was the question?
Sorry, difficulty in a clinical trial.
Yeah, Dennis. So that's actually part of the reason we're based in Oxford, because as I'm sure a lot of you are aware, Oxford have a lot of experience in clinical research. The other thing, so you are completely right, it is a very sensitive area because there is a baby there, and everyone, I think, panics when you think about pregnancy and babies and thalidomide. The beauty of using mitochondria is, like I said on my slide, it is 100% the mother's. So you're not giving something foreign. It is a completely autologous donation if it comes from the mother. So we've already had talks with MHRA and CTRG about clinical trials. And I mean, there will still be lots of hurdles, but actually from a scientific basis, hopefully some of these will be overcome.
Sure, great comments. Thank you very much. Because this session is kind of conceptual to bridge Japan and the UK. And there's many good Japanese startup company want to do global, and I believe UK is a great place to start business with. And also we've done a lot of startup technologies from UK to Japan for the another soft landing, but the Japan to the UK is more that we are facing to some issues, and we have to do many things as well. So this links to the yes, same to yours, but the question from Isabel. It's common question to everybody, but have you faced any particular obstacles with your UK legal entity setup?
Yeah. So yeah, the question about setting up a UK entity.
Yep.
I think it would be a lie if I said there were no obstacles. There's always going to be obstacles. Interestingly, the biggest obstacle for us was setting up a UK bank account with all the due diligence checks. It just took a lot longer than expected. We were very lucky from a collaboration side and the legal side in that I've worked at Oxford before, and you can probably tell from my accent that I have lived in the UK before. So we had, I think it's very important to have someone on the UK side, especially on the collaborator side, invested. So I had professors at the University of Oxford who wanted to collaborate with us. So that made the research collaboration a lot easier. I would just say, and I don't know if Dr. Matsunaga, you agree, is I think it's all about expectations. So I think if you expect that there are going to be obstacles, then at least it won't come as a shock. I think, especially for startups, you want everything to happen quickly. But if you have too high an expectation, especially international collaborations, there will be, obstacles. A lot of it is cultural. So I think as long as you have that kind of expectation, then, hopefully you will get to the goal that you want to.
Cool, thank you. And in addition to that, I think the IT government sector is very helpful when you want to do that. Right, so it's already 10:58, so I will hand over to Eileen, One Nucleus, but thank you again for the great presentations, and thank you very much for the audiences for keeping listened for the last 60 minutes. If you can open the videos, I can maybe put the screenshot.
I think that would look nice.
Yes.
And thanks very much to everyone who presented, because they were all very interesting technologies with really good potential. Thank you for the Space BD. I now have more insights on how it works, and I think it's a really cool idea. But yeah, to your point, Matashi, I think it's, and that relates to the last question we had about setting up in the UK. I think there are different levels of collaboration with the UK, whether it's having business development relationship, and I think you're very well placed to support that, and we're happy to help too, and I hope this platform can help as well. But if it's about setting up in the UK, yes, there are structure like the IT or even regional structure that can be really, really helpful in all these very practical things of opening bank account, because when you're an entrepreneur, your focus should be on your science and raising money, probably not really on all these practical things. So there are people to help. But anyway, that was really good to have you. I really enjoyed the session. As I said, it was recorded. Thank you for everyone who attended. You can rewatch it, share with your colleagues, and that will be available on the Genesis app. And obviously make contact with them on the app. It's still open until the 7th of January, so you can organize, meeting afterwards or just chat. So thanks, everyone.
Thank you, Ali. Bye. Thank you, everyone.
Bye.
Thank you.
Thank you.